Good morning, everyone. This is Nicole Ali. So, I've been really busy working on a few projects and analyzing a lot of data. So, please forgive me for the delay on videos, but I'm back at it again. You know, you can hear the music in the background. Rich Homie Quan, the song Differences. It's the instrumental, but if you know the song and you know based on the title and a lot of things going on, you already know what it's going to be about. So the difference between those with high IQs and those of us of the royal family is there are certain things that we will do and there's certain things we will not do. This is a two-part video. The first part is going to be basically talking about this particular kit. You see the price right there, $445, which is relatively cheap compared to a lot of the kits that we use. Some kits go up to $2,000, $3,000 a piece. And the purpose of this kit, as you can see, these are the tubes, the buffers, the reagents that you use to detect glutamine and glutamate in your assay, in your sample, biological samples. Now, these biological samples can be serum, plasma, tissue culture medium. You can detect it in cells. So you can perform this performance experiment on a variety of sample types, as they say here. Also helps you determine the viability of your sample as well. It's a bioluminescent assay for the rapid and sensitive measurement of glutamine and glutamate from different samples. You can detect signal interference. It helps to eliminate signal interference. And because some colorimetric or fluorescent assays don't allow you to do that. You can detect glutamine and glutamate. So what is glutamine and glutamate? Glutamine is an amino acid required for you to conduct metabolic processes. Glutamate or glutamic acid is a byproduct of the biosynthetic process of converting glutamine into glutamic acid or glutamate. You need an enzyme called glutaminase in order to convert glutamine into glutamate. They give their titration curves, so that helps you to detect glutamine, seen in the blue line, and glutamate, seen in the yellow line. I actually conducted this assay yesterday on a sample of tissue culture serum containing media. The cells were treated with a drug for prostate cancer, an anti-androgen. And after 72 hours, I did the analysis. So you're not looking at my data, you're looking at data from their website, which shows you glutamine consumption and glutamate secretion by the SCOV3 ovarian cancer cells. So this is used in several different types of cancer cells as well. Now, you hear the song changing, right? Because I'm going to leave a link to this in the description box, of course, so you can read it at your leisure. Understand how this system works, how this processing system works for determining glutamine and glutamate. Now, how do these people with high IQ use something like this? Because I'm only detecting this in cells and in the media that the cells are eating, right? There's no money involved. There's no nefarious activity involved. I'm just trying to determine if this drug is reducing the ability for the cells to go through metabolism. And I think that the drug is actually helping the cells metabolize more. I haven't analyzed the data. I just ran an experiment yesterday. But you have a group of people with high IQs that feel that cash rules everything around them. Right? So they're using glutaminase. Remember, that's the enzyme that helps to convert glutamine into glutamate. And you probably have heard of this topic. This is the second part of the video. Meat glue. What it is and what you should know about. This comes from a website called Deliciously. They published it earlier, updated August 26, 2018. This woman, Kate P., is a dental hygienist, pyrography artist, avid gardener, writer, vegetarian, world traveler, and many other things. So she thought it was important to talk about meat glue. This is a picture of a piece of meat that has been literally glued together with this product called meat glue. The subtitle says meat glue has lots of application. Lots of applications, lots of ways you can use it. You have maybe seen these viral videos going around about meat glue. It's deceptive, it's nasty, and it's potentially deadly. I couldn't agree with her more. Read on to learn more about why it's used, what it is, and the dangers it poses. I'm going to go ahead and cut through the bullshit. Meat glue is glutaminase. The same enzyme I just talked about, they call it frankenmeat. You know, it's an industry standard. The chances are if you eat meat or even tofu, you're consuming a binding agent on a monthly, if not weekly basis. And for all you vegetarians and vegans out there, please note this stuff is not friendly to you and might be in your cheese, dairy, or tofu. Meat glue is scientifically known as transglutaminase. Its brand name is Activa. Activa is managed by the Ajinomoto company. I'm assuming they are Japanese. The same folks who brought us the pervasive neurotoxins aspartame and MSG, monosodium glutamate. Glutamate is the acid I just talked about previously. So I'm showing you the connection between the laboratory work I do and how we're trying to determine how certain drugs used in cancer patients can be helping the cancer cells do better metabolically, which is why we feel it necessary to incorporate alternative forms of therapy to help these patients. There's little debate within scientific circles as to the neurotoxic effects of these food additives. The jury's still out about transglutaminase, but if you ask me, anyone willing to sell neurotoxins to the public cannot and should not be trusted. I agree. Transglutaminase, also known as TG. There's TG 1, 2, 3, 4, 5. And those are the ones I know about, but I know there's plenty others. It's an enzyme that helps proteins bind permanently together through the formation of covalent bonds. Covalent bonds are very hard to break. Usually with heat or pressure or radiation, you can break covalent bonds. You know, radiation, this is a Japanese company. Fukushima, anybody? So the TG forms cross-linked, insoluble, and irreversible protein polymers. So what I just said about breaking it with heat, pressure, or radiation, yeah, you can break the bonds. But if you leave these materials in close contact, they actually form bonds back again together. And the second time, the bonds are harder to break. It's insoluble, meaning that it cannot be dissolved in water. It cannot be dissolved in a solution, a solvent. You have natural examples of these stable polymers in blood clots, your skin, and your hair. That's why your hair doesn't fall apart when you pull a brush or a comb through it. You also have chemicals proteins such as collagen, integrin, keratin that keep, these polymers together as well. So you have the chemical basis is a protein-bound lysine. Lysine is another amino acid. Protein-bound glutamine, another amino acid. So you have a free amine plus a carboxamide. This is an example of a package of Activa RM, 2.2 pounds, 1 kilogram. Ajinomoto, it says a product of France on the package. This is what it looks like. Transglutaminase looks a lot like cocaine or powdered sugar. It's a white powdery substance. That's what it looks like. Okay. They make it from cultivating it from bacteria using blood plasma from pigs and cows. Some TG is made from cultivating bacteria using vegetable and plant extracts. Most TGs are mixed with other ingredients, including gelatin. Gelatin that usually comes from pigs, quote unquote healthy female pigs that are lactating or are in heat, meaning that they are of a reproductive age. They also use caseinate, which is a milk derivative from cows. The issue, manufacturers don't have to tell you which method they're using, what type of animal plasma they use, cow or pig, or anything else that most of us would like to know more about. As with most things, the only way you really know what you're consuming is if you make it yourself. Depending on how stringent a vegetarian you are, this may or may not ring some bells for you. TG is often used to increase yields of tofu production. Vegans avoid this stuff like the plague. I bet they do. So Judaism and pork products don't go together. So make sure you're buying kosher cuts. If you can buy kosher cuts. Even with the kosher beef or kosher chicken, you have to make sure that that is not having glutaminase in it as well, because if they send it to a second party to process, they could add glutaminase in there. Very small quantities, but enough of it over time can affect you health-wise. And they give you an example of how you spin down blood. 55% of it is plasma. Small amount, less than 1%, is white blood cells and platelets. The red blood cells make up 45%. So when we do the analysis, this is how on plasma or on serum, this is how we spin it down, and it looks like this in a test tube. It separates like that. And there's a video here about meat glue. This guy stupidly ate some of it after it was prepared. I'll leave this link in the description box. I hope this was informative to you all. Please watch what you're eating. This piece of meat here looks like a lamb chop, or pork cuts. So they're pork cuts that they've bonded together with meat glue. Once the animal is dead, of course, it doesn't matter how you process it. To them, they feel like, well, we can process it any way we want. This is a picture of seafood that's been sliced and bounded together. So the outside red part looks like tuna. The inside pink part is probably salmon, and they've bonded it together with meat glue. What they do is add the powder in between the pieces they want bonded together. They roll it up in plastic wrap, then in foil, to increase the pyrogenics of it, meaning that it's thermogenic, so it has heat, and that heat bonds the molecules together, the proteins together. This is an example as well of lamb and scallops. So they've taken lamb, which is in red, uncooked lamb, and bonded it to scallops, which is white. Then you can go and slice that, and you have lamb and scallops mixed together. And then they show you the points of fusion. This looks like it's supposed to be beef. Restructured steak cuts. Those of you who like to grill and eat steak, you might want to watch what you eat, and you might want to find out where the meat is coming from and what's going on with it. It could be bonded. So they also tell you what it's used for. Binding meat improves the mouthfeel, water retention, and appearance of processed meats. Used in molecular gastronomy. Thickens egg yolks, strengthens dough mixtures, thickens dairy products like yogurts and cheeses, increases the yield in tofu production. Everybody knows what this is: chicken nuggets. Chicken nuggets is everything except what you should be. It's everything that you should not be eating. All the good parts of chicken that you want to eat, this is everything that's not there. You know, the beak, the eyes, the fatty pieces, the diaphragm, anything that you wouldn't want to eat. Chicken feet, all that is ground up and made into chicken nuggets, and now they can glue them together. Even chicken tenders are used. They use meat glue on them. Imitation crab meat, now they can bind it together, and it's not even really crab. The FDA classifies it as generally recognized as safe. So you can see how they feel about it. They don't give a damn, because it's all about that money. The USDA approves the use of neurotoxins like MSG, aspartame, fluoride, pesticides, Monsanto, and herbicides and growth hormone, antibiotic injected meats. So these are microscopic pictures of bacteria in your body. And please believe, this meat glue is not necessarily sterile. You know, you're going to be feeling like this dude right. You're going to be feeling like you got to go boo-boo. You're going to be feeling like, wow, I don't feel well at all. So try to avoid that, and, you know, learn about what you're eating, learning about what they're putting in your food. You know, just be more aware of it. I hope you all enjoyed this video. I hope it wasn't too long. I'm just trying to put more information out there about what these things are. And remember to like, comment, share, subscribe. Realize that I'm doing the research myself, trying to study the metabolic interactions and metabolic promotion of cancer cells using standard therapy on the market right now. I hope that you all can find some enlightenment in my videos. Send me videos and stories to my email. I'll leave that in the description box or pinned comments, and I'll talk to you in the next one.